Hello and welcome to Sam Biology class. Friends, today we shall discuss about DNA footprinting. DNA footprinting is a technique in molecular biology by which we can determine the sequence specificity of DNA binding proteins. So this technique is used to study the interactions between DNA and proteins both within and outside cells. DNA footprinting was developed by David J. Gallas and Albert Mitz in 1978 in order to study the binding specificity of LARC repressor protein. This method is based on the chemical sequencing of Maxam and Gilbert. The regulation of transcription has been studied extensively, but yet there is much to be known. Transcription factors and associated proteins that binds to promoters, enhancer or silencers to drive or repress transcription is the fundamental to the understanding of unique regulation of individual genes within the genome. By DNA footprinting, we can identify the location on the DNA where a specific protein binds and interacts. This technique is used to study the interactions between DNA and specific proteins in order to understand the mechanism of gene regulation. In footprint analysis, there are four main steps. They are labeling, incubation, cleavage and electrophoresis followed by photoradiography. First of all, the DNA template to be studied is amplified by polymerase chain reaction. After amplification, the DNA template is labeled at either 5' or 3' end depending on the location of protein binding site. The labeling can be radioactive or fluorescent. Radioactive labeling has traditionally used for footprint analysis because it is very sensitive and is optimal for visualizing a small amount of DNA. Fluorescent labeling is a desirable advancement because it do not involve the use of radiochemicals. After labeling, the DNA templates are divided into two sets of sample. One set of sample is not incubated with protein, whereas the second set of sample is incubated with specific protein so that the protein will bind at a particular region on the DNA. By this technique, we want to identify the exact location on the DNA where the specific protein binds. Next step is cleavage. Cleavage can be performed by using several agents. A suitable cleavage agent should be sequence neutral and it should be easy to use and to control. Normally, DNase 1 is used for cleavage. This enzyme is a double-strand endonuclease. It binds at the minor groove and cleaves the phosphodiester backbone of DNA, thereby generating a series of fragments. The cleavage is performed under specific conditions so that DNase 1 will make only one cut per molecule. When cleaved, the first set of DNA template which is not associated with protein will give rise to a continuous ladder which differs by one base only. But the second set of DNA template, because it is associated with protein, so its cleavage will be interrupted because the region where the protein has been bound will be protected from cleavage. So there will be a gap. This gap is called as DNA footprint. After cleavage, The fragments are separated by polyacrylamide gel electrophoresis in separate lanes along with sequencing ladder so that it will be possible to identify the location and sequence where the protein has been bound. Here in the gel you can see that there are 6 separate lanes. The first lane contains the bands which are formed due to the cleavage of the DNA template which is not associated with protein, so there is a continuous ladder. Whereas in the second set of sample which is associated with protein, because the cleavage is interrupted, so there will be a gap. This gap is called as DNA footprint. By analyzing the bands formed by DNA sample which is not associated with protein with the bands that are formed by the DNA sample which is associated with protein along with sequencing ladders, you can identify the exact location where the protein has been bound to the second set of DNA template. To identify the location, the gel is subjected to autoradiography which enables us to identify the protein binding site. So friends, this is about DNA footprinting. By this technique, we can study the interaction between DNA and different type of proteins that help us to understand the mechanism of gene regulation. So friends, thank you very much. Please subscribe my YouTube channel, Sam Biology Class.